A very good evening to all. So today we are going to discuss some of the questions which came in test. Let's start with question number 1. Question 1 says, read the following statement and select the correct option that bears the true statements. Statement number 1, during the S phase, the amount of DNA per cell doubles, but the chromosome number remains same. Statement 2 says that a cell in G0 phase is metabolically inactive but no longer proliferates unless called on to do so depending upon the requirement of the organism. Statement 3, cell division is a progressive process that shows the distinct boundaries between the various stages. 4 statement says at the end of prophase, Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope get disappear. So options are given. Quickly answer what you think what will be the correct option for this question. So statement 1 says that during the S phase, amount of DNA per cell double. Yes, during the S phase, DNA duplication occurs, but the chromosome number remains same. Yes, chromosome number remains same, but the amount of DNA get doubles during the S phase. This statement is true. Second says a cell in G0 phase is metabolically inactive. No, the cell in the G0 phase is metabolically active, but no longer proliferate unless called on to do so depending upon the requirements of the organisms. So this statement is wrong because a cell in G0 phase is metabolically active. Statement number 3 says that cell division is a progressive process that shows the distinct boundaries between the various stages. Cell division is a progressive process, yes. That shows the distinct boundaries, no beta. Though the cell division is a progressive phase, but the distinct boundaries between the stages cannot be made. Okay. So distinct boundaries cannot be drawn between the various stages. Fourth is at the end of prophase, Golgi complex, endoplasmic reticulum, nucleolus, and nuclear membrane, they get disappear. True. These are the characteristics of the prophase because during the prophase, Golgi complex, endoplasmic reticulum, nucleolus, they get disappear. Disintegration of nuclear membrane or envelope takes place and the condensation of the chromatin occurs in the prophase. And the question is asking select the correct option that bears the true statement. So statement 1 is true and statement 4th is true. So correct option is D. Only 1 and 4th. Question number 2 says splitting of centromere and separation of chromatid occurs during. Option A is anaphase of mitosis. Option B is anaphase of meiosis 1. Option C is anaphase of meiosis 2. And option D is both A and C. What do you think? What will be the correct option for this question? They're asking about the splitting of centromere and separation of chromatid. So the question is saying the splitting of centromere and the separation of chromatid occurs during in mitosis, anaphase of mitosis, sister chromatid, they get separated. And they start move towards the opposite pole. We know that meiosis is divided into meiosis 1 and meiosis 2, right? So anaphase of meiosis 1, in anaphase of meiosis 1, homologous chromosome, they get separated. Remember this, it's very important. And in anaphase 2 of meiosis 2, sister chromatid gets separated. Meiosis 2 and mitosis are same. Okay? Remember this. Meiosis 2 is also equational division and mitosis are also equational division. But meiosis 1 is a reductional division. And the question is asking the splitting of centromere and the separation of chromatid occurs during anaphase of mitosis and anaphase of meiosis 2. So both A and C. So the correct option is D, both A and C. Next question says, which one of the following option gives the correct characterization of the 6 plants on the basis of the position of ovary? So they have given the superior ovary, half inferior ovary, and the inferior ovary, and they have given the plant's name. So you have to match it. First one is superior, then half inferior, and inferior. In superior, the gynoecium very good, Sammy. So in superior, the gynoecium occupies the highest position. If this is gynoecium, the other whorls of the flower, that is sepal, androecium, stamens, petals, they are all arises from the so the gynoecium is situated above and all the whorls or the other parts of the flower, they arises from below the ovary. Okay. So this is a condition of superior and the flower is known as a hypogynous flower. And the example of superior is brinjal, China rose, and mustard. In case of half inferior, the gynoecium occupy the center position. And the other parts of the flower as sepals, petals, and the stamens, they are arising from the rim of the thalamus at the same level. You can see this at the same level. So this type of ovary are known as half inferior, or we also call it half superior. And the example of half inferior are rose, peach, and plump. In inferior, the ovary is enclosed by the thalamus. And the other part of the flower, just like sepals, petals, they are all arising, they all are situated above the ovary. So this type of arrangement, we call it as inferior ovary. And the example of inferior are guava and cucumber. Please remember the example. These are very important. Very good, Veena. Now you can easily answer. Option 1 says that superior is apple, datura. Half inferior says they plump and peach. Inferior, they says mustard. Mustard and China rose, they are superior, not inferior. B, superior mustard, China rose. Half inferior, plump and peach. Inferior, guava and cucumber, absolutely correct. So the correct option is B. But let's see this option C and D as well. Here they say that superior is peach. Peach is half inferior. So this is also wrong. Option D says superior, onion and rose. Rose is half inferior. It's not superior. So this is wrong. So the correct option is option number B. Now moving next. Question 4 says, choose the correct statement. Statement 1 is pyrenoids. They are present in all algae that is meant for storage of starch only. Second statement says that there is a gelatinous coating of algin is found on the cellulosic wall of Phaeophyce. Statement 3 says rhodophyce contain R phycoerythrin pigment as well as 2 and unequal lateral flagella. Statement 4 says majority of red algae are marine with greater concentration found in the warmer areas. Quickly answer what you think, what will be the correct option for this question. Statement 1 is saying that pyrenoids are present in all algae. No, pyrenoids, they are present in green algae. In most member of green algae. That is meant for storage of starch only. They store protein besides starch. Statement 2 says, gelatinous coating of algin is found on the cellulosic wall of Phaeophyce. Phaeophyce is a brown algae. This is correct. Because the vegetative cells of the brown algae, they have the cell wall, and around the cell wall, there's a gelatinous coating of algin is present. Statement 3 says, rhodophyce contain R phycoerythrin pigment. Yes, because of which the rhodophyce are the red algae, and because of the presence of R phycoerythrin, the red algae appears red in color, as well as 2 and unequal lateral flagella. No, in red algae, the flagella are absent. Remember this. In red algae, the flagella are absent, and these 2 unequal lateral flagella, they are the characteristics of brown algae, Phaeophyce. Statement 4 says the majority of red algae are marine with a greater concentration found in the warmer area. This is correct. The majority of the red algae, they are marine. And the red algae, you can see the red algae are present on the surface of water also, and they can be present in the depth of the sea also. So the question is asking about the correct statement. Statement 1 is not correct. Statement 2 is correct. Statement 3 is not correct. And statement 4 is correct. So 2 and 4th. So correct option is option number B, which says 2nd and 4th. I hope it is clear to everyone. Moving next, consider the following statement 1, 2, 3, and 4th and select the right option for the 2 correct statement. Statement 1 says the Z scheme of light reaction takes place in the presence of PS1 only. Statement 2 says only PS1 is functional in cyclic photophosphorylation. Statement 3 says cyclic photophosphorylation results into synthesis of ATP and NADPH2. Statement 4 says stroma lamellae membrane lack PS2 as well as NADP reductase enzyme. You have to tell which statements are correct. What do you think? What will be the correct option for this question? Very good, Swami. So now look at the statement. Statement 1 says the Z scheme of light reaction takes place in the presence of PS1 only. No. You know that light reaction, it includes two things. One is a cyclic photophosphorylation. And another one is non-cyclic photophosphorylation. Cyclic photophosphorylation involves only PS1. And non-cyclic photophosphorylation, which also gives the Z scheme, it involves both photosystem 1 and photosystem 2. In cyclic photophosphorylation, only ATP is formed. But in non-cyclic photophosphorylation, oxygen is evolved, ATP is formed, and NADPH is formed. Remember the product of the light reaction. Okay, now look at your statement. Statement 1 says the Z scheme of the light reaction takes place in presence of PS1 only. No, the Z scheme, that means the non-cyclic photophosphorylation, it includes both PS1 and PS2. So statement 1 is wrong. Now statement 2 says that only PS1 is functional in cyclic photophosphorylation. Yes, in cyclic photophosphorylation, only PS1 is functional. So statement 2 is correct. Statement 3 says cyclic photophosphorylation result into synthesis of ATP and NADPH2. No, in cyclic photophosphorylation, only ATP is formed. Whereas in non-cyclic photophosphorylation, oxygen is evolving because in at photosystem 2, splitting of water takes place. And because of the splitting of water, oxygen is evolved. So in non-cyclic photophosphorylation, oxygen is evolved, ATP is synthesized, and NADPH is formed. So statement 3 is also wrong. Statement 4 says that stroma lamellae membrane lack PS2 as well as NADP reductase. Yes, stroma lamellae, they lack PS2 and NADP reductase. In stroma lamellae, this PS1 is present and this cyclic photophosphorylation occurs. And the question is asking about the correct statement. So statement 2 is correct and statement 4 is correct. So the correct option is B. I hope it is clear to everyone. Now question number 6. Study the following statement regarding the chemiosmotic hypothesis in mitochondria and select the correct ones. Statement 1 says that F1 headpiece contains the site for the synthesis of ATP from ADP and inorganic phosphate. Statement 2 says that F0 part form the channel through which the proton crosses the inner membrane. Statement 3 says for ATP production, proton pass through the F0 from the matrix into the perimitochondrial space due to electrochemical proton gradient. What do you think? What will be the correct option for this question? There's an enzyme which ATP synthase. This enzyme has two parts. There's F1 and the F0. F0, it forms the channel, and the F1 part, it has the catalytic activity. This F0 part, it is embedded in the inner membrane because it is forming the channel. And this F1, it protrude out in the matrix. Like this. If this is a membrane, inner membrane, this is F0 and this is F1. And this F1 is responsible for converting this ADP and inorganic phosphate into ATP. So statement 1 says that F1 headpiece contain the site for the synthesis of ATP from ADP and PI. Yes. F0 part forms the channel through which the proton crosses the inner membrane. Yes. This is the space in intermembrane space, and this is a matrix of mitochondria. Okay. So when the electron, they move from one electron carrier to the another, in return, the proton from the matrix, they are transferred to the intermembrane space. So the intermembrane space, they have a high concentration of the protons. So when these protons, they pass through this F0 channel, the proton gradient breaks. And when it breaks, it generates an energy, and this energy is utilized by this F1 headpiece, and it results in the synthesis of ATP. Okay, so the high concentration of the proton are present in the intermembrane space. For the ATP production, the H protons, H plus ions, pass through the F0 from the matrix into the perimitochondrial space due to the electrochemical proton gradient. So for the ATP production, this H plus pass through the F0. Yes, the H plus protons, they will pass through the F0 from the matrix to the intermembrane space or the perimitochondrial space. No, from the intermembrane space or the perimitochondrial space toward the matrix. Okay, so this is a wrong statement. So only statement 1 and 2 is correct. So the correct option is A. That is only 1 and 2. I hope it is clear. Now moving next. Choose the correct sequence of development of female gametophyte. Option A says megaspore mother cell, then megaspore tetrad, megaspore, then nucellus, and female gametophyte. Option B says nucellus, megaspore mother cell, megaspore tetrad, functional megaspore, and then female gametophyte. Option C says megaspore tetrad, megaspore mother cell, megaspore nucellus, and female gametophyte. The option D says megaspores, megaspore mother cell, megaspore tetrad, nucellus, and female gametophyte. What do you think? What will be the correct option or the correct sequence of the development of female gametophyte? One cell of the nucellus get differentiated into megaspore mother cell. This megaspore mother cell will undergo meiosis and it will produce the 4 haploid megaspore. Or we can also call it as megaspore tetrad. Out of this tetrad, 3 will get degenerated and only 1 remain functional. Now this functional megaspore will undergo mitotic division and it will form the female gametophyte. Which we also call it as embryo sac. And we call it as female gametophyte. Gametophyte means the plant body which bear the gamete. So embryo sac, they have the female gamete, which is egg. So that is why we also called embryo sac as female gametophyte. First is nucellus, then megaspore mother cell, then megaspore tetrad, then functional megaspore, and female gametophyte. So the correct sequence is, or the correct option is option number B. I hope it is clear to everyone. Now the question 8 says in E. coli, the operator gene combines with, option A is inducer gene to switch on the structural gene action. Option B is inducer gene to switch off the structural gene action. Option C is regulator protein repressor to switch off the structural gene action. Option D is regulator proteins to switch on the gene action. What do you think? What will be the correct option? In the previous class, we have discussed the lac operon. This is a promoter for regulatory gene. This is the regulatory gene. The lac promoter. This is the operator. And these three, Z, Y, A, they are the structural gene. So this regulatory gene will form the mRNA. And this mRNA will form the protein, which is the repressor. When the inducer is absent, this repressor will bind to the operator. When this repressor will bind to the operator, it does not allow the RNA polymerase to bind with the promoter. So the transcription of the structural gene will not take place. So in this case, the transcription of the structural gene will not take place. So in this case, the operon will be switched off. Now look at the question. In E. coli, the operator gene combined with inducer gene? No, operator gene does not combine with inducer gene. Inducer gene to switch off the structural gene action? No. Regulator protein repressor to switch off the structural gene action. That is the correct option. Option D says that regulator protein to switch on the gene action. When the regulator protein bind with the operator, the RNA polymerase cannot bind with the promoter. So the transcription of the structural gene will not take place. So it's switch off, not on. Okay. So this is option D is also incorrect. So the correct option is option number C, that the regulator protein, which is repressor, binds to the structure, bind to the operator gene. Okay, and it leads and it result in the switch of the structural gene action. I hope it is clear to everyone. Now moving next. Question 9 says, how many of the following statements are correct? Statement 1 is the number of RBC in human female is 5.5 to 6 billion mm. Statement 2 says that WBC are the most abundant cells of blood. Statement 3 says that humans RBC is oval and non-nucleated. Statement 4 says the average lifespan of RBC is 120. What do you think? What will be the correct option? You have to find out the correct statement. Statement 1 says the number of RBC in human female is 5.5 to 6 billion. The number of RBC in human female is 4.5 to 5 million. MMQ. Very good, Sammy. Statement 2 says WBC are the most abundant cells of blood. The most abundant cell of the blood is RBC, then platelets, and last is WBC. WBC are 6,000 to 8,000 mmQ. RBC, they are 5 to 5.5 million. Platelets, they are 1.5 to 3 lakh. So the most abundant cell of blood is RBC, not WBC. So second is wrong. Third says that human RBC is oval and non-nucleated. Human RBC is biconcave. So third is also wrong. Statement 4 says the average lifespan of RBC is 120 days. Yes, this is correct. The average lifespan of RBC is 120 days. And the question is asking how many statements are correct. So only one statement is correct. So the correct answer is A, that is 1. Question 10 says that find out the incorrect statement. You have to find out the incorrect statement. A says that in heart attack, the heart muscles are suddenly damaged. B is 130 to 90 is the hypotension. C says that heart failure and the cardiac arrest are different. And D is angina pectoris is identified as acute chest pain. What do you think? What will be the correct option? A is saying that in heart attack, the heart muscles are suddenly damaged. Yes. In heart attack, the heart muscles are suddenly damaged due to the inadequate supply of blood. (130 to 90 is hypotension? No. Hypotension or the low blood pressure is when systolic pressure is 90 and diastolic is 60. And hypertension, which is a high blood pressure, 140 to 90, where systolic pressure is 140 and diastolic pressure is 90. He said the heart failure and cardiac arrest are different. Yes, heart failure and cardiac arrest are different. Cardiac arrest when there's a complete stoppage of the heartbeat, and heart failure when the heart failed to pump enough blood to the rest of the body parts. Angina pectoris is identified as the acute chest pain. Yes, this is also correct. And the question is asking, find out the incorrect statement. So the incorrect statement is B. And a correct option is also B with respect to the question. Moving next, when a neuron is not conducting any impulse, that is resting, the axonal membrane comparatively dash to potassium ions and nearly dash to sodium ions. Option A says less permeable, impermeable respectively. Option B says more permeable, impermeable respectively. Option C says less permeable or more permeable respectively. And option D says impermeable and impermeable respectively. What do you think? What will be the correct option? Very good, Swami. So during the resting, when the membrane is in the resting state, you observe that the membrane is more permeable to potassium ions and impermeable to sodium ions and negatively charged proteins. Because of which, in the resting state, we observe that positive charge is more on the outer side. On the inner side, there's a negative charge because the membrane is impermeable to negatively charged proteins. So sodium are more in concentration outside and potassium are more in concentration inside. This state we call it a polarization. But when this neuron receive a threshold stimulation, in that case, the membrane becomes permeable for sodium ions. And the condition is known as depolarization. Now the sodium channels, they get open and the sodium from outer, they move inside. Now the positive charge is on the inner side and negative on the outer side. And the question is asking, when the neuron is not conducting any impulse, that means when the resting state is present, in that condition, the axonal membrane comparatively more permeable to potassium and impermeable to sodium and negatively charged proteins. So more permeable to potassium ions and nearly impermeable to sodium ions. So the correct option is B. Question 12 says that during the muscle contraction, first is that I band gets reduced, second A band retain the length, third length of the sarcomere decreases, fourth breakdown of ATP, So if this H zone remains unchanged, what do you think what will be the correct option? They're asking about the correct statement. When the muscle is in the resting state, that means there's no contraction. These are the Z lines. Now see, this is the I band. From here till here is the A band. And this is the H zone or the Henson zone. H zone is the portion where no actin filament or the thin filament are present. There's only the thick filament, which is myosin. And the A band, it includes both actin filament and the myosin filament. You can see this, the actin filament, they are overlapping the thick filament of the myosin. And this I band, they only contain the thin filament, that is actin filament. This is the resting state when there's no contraction. But when the muscle contract, you can see this. Now the H zone is completely disappear. During the contraction, the H zone disappears. The I band, it starts reducing. Here you can see the length of the I band, and now the I band becomes short, so it reduced. But the length of the A filament remains as it is, because the length of the A filament represents the length of the thick filament, which is myosin, which remain constant. Okay, remember that's very important. And the portion between the two successive Z line, it represents the sarcomere. And the length of the sarcomere also decreases during contraction. So remember, during muscle contraction, I band get reduced, length of the sarcomere decreases, and the A band retains the length, and the H zone disappear. And for the contraction, ATP is required. So First is correct, second is correct, third is correct, fourth is correct, but fifth is not correct. Because here they're saying the H zone remain unchanged. No, the H zone first is decreasing and later it disappear because of the contraction. Because now these actin filament, they have overlapped each other. They are overlapping the thick filament of myosin. So B is the correct option. Like only 1, 2, 3, and 4 are correct. Moving next is which of the following statement are true, false? 1. Thyrocalcitonin regulate the blood glucose level. Oxytocin stimulate the contraction of uterine muscle during birth. Grave disease is caused by the malfunctioning of the thyroid gland. ADH stimulate the absorption of water and increase the urine production. So you have to answer whether the statement is true and false. Quickly answer what you think, what will be the correct option. Thyrocalcitonin. It is produced by a gland, thyroid gland. And the main function of this hormone is to regulate the calcium level in the blood. So statement 1 is saying that thyrocalcitonin regulate blood glucose level. No, it regulates the calcium level in the blood. So this is false. First is false. Second says oxytocin stimulate contraction of uterine muscles during birth. Yes, it's true. Third says grave disease is caused by malfunctioning of thyroid gland. Yes. Grave disease is an autoimmune disease and it occurs due to hyperthyroidism. Hyperthyroidism, when the thyroid hormone like T3 and TH, they are produced in a very high concentration. Excess secretion of T3 and T4 lead to a condition which is known as hypothyroidism, and hypothyroidism lead to a disease which is known as grave disease. So statement 3 is true. Statement 4 is that ADH stimulate the absorption of water and increase the urine production. ADH, another name of ADH is vasopressin. And the function of ADH or vasopressin is reabsorption of water and electrolyte. And they reduce the loss of water through urine. So they will decrease the urine volume. But here they're saying increase the urine production. They decrease the urine production. So this is also false. So statement 1 and 4 are false. Statement 1 and 4 are false. And 2nd and 3 are true. So the correct option is C. I hope it is clear to everyone. Next question says, match the organism with its use in biotechnology. Column 1 contains Bacillus thuringiensis, Taq polymerase, Agrobacterium tumefacium, and PBR322. Column 2 contains cloning vector, tDNA, Thermus aquaticus, and the cryoprotein. You have to match the column 1 with column 2. Quickly answer what you think, what will be the correct option for this question. A is Bacillus thuringiensis. Bacillus thuringiensis is a bacteria that contains a cry gene, and the cry gene is isolated from this bacteria and inserted into a plant like beet. And genetically modified plants are obtained like BT cotton and BT cone. So this cry protein, it provides the resistance against the pest. So A, cry protein, or S. B, Taq polymerase. Taq polymerase is a special type of DNA polymerase which is used in PCR, polymerase chain reaction. And the function of Taq polymerase is polymerization. And it is a thermostable enzyme. And it is isolated from an organism named as Thermus aquaticus. So BR. C is Agrobacterium tumefacium. It causes the crown gall disease or the tumor disease in plant cell. And this Agrobacterium tumefacium, it has the TI plasmid. So it acts as a cloning vector. So CP. And D, this Agrobacterium tumefacium, they have a tDNA. We also call it as transfer DNA. So CQ and PBR322, it's a plasmid. This is a plasmid. It is an artificially synthesized plasmid. And we know that the plasmid, they act as a cloning vector. So D is P. AS, BR, CQ, and DP. So the correct option is A. Next question says, which of the following tools of recombinant DNA technology is incorrectly paired with its use? A says restriction enzyme production of blunt end. B says DNA ligase creates sticky end of restriction fragments. C says DNA polymerase used in a PCR to amplify the section of DNA. D says reverse transcriptase production of cDNA from mRNA. Question is asking which of the following tool of recombinant DNA technology is incorrectly paired. So there are two types of restriction enzymes. One are exonuclease and another one are endonuclease. Exonuclease which cut the DNA from the terminal ends and endonuclease which cut the DNA within. Endonuclease are it further produces two types of ends, sticky ends and blunt end. Another name of sticky end is staggered end, overhanging ends, or the cohesive ends. And another name for the blunt ends are flush end. So option A says the restriction enzyme production of blunt ends, yes. B says DNA ligase creates sticky ends of restriction fragment, no. DNA ligase, they function as molecular glue. They help in the joining of the vector DNA and the foreign DNA. DNA polymerase used in PCR to amplify the section of DNA. Yes, the special type of DNA polymerase which is used in PCR is a Taq polymerase. So this is also correct. Reverse transcriptase production of cDNA from mRNA. Yes,